Thank you for spending your precious time with me. I'm truly honored and excited to present you the utilization of microbes in vegetables at BEVO Farms. So that's the topic today. So for a quick introduction, the table content, we're going to introduce the what do BEVO Farm do, what is microbes, disease triangle and life form triangle, interaction of plant pathogens with susceptible hosts, symbiotic relationship of plant with beneficial microbes. And then we will jump straight to objective and why is BEVO using microbial. And then the new methodology that we introduced last year during pepper season, and then the new releasing method basically that we'll be covering that part. And then we will take a quick break. And then after that, will be how do we detect, like when we release that, how do we detect the things that we release? Because they are so small, we can't really see them. So we're just gonna see the observation of the pepper crop, cucumber, and strawberry. And then the future conclusion, acknowledgement, and references. So what does BEVO do? BEVO, we grow veggie all year round, particularly on cucumber, pepper, tomato, eggplant, strawberry, and etc. Bedding crop as well, all year round, hydrangea, chrysanthemums, poinsettias, orchid, mixed basket, and etc. etc. We have 3 different locations. In BC, we have 3 different locations. BEVO Langley, Pitt Meadows, Eldergrove, and in Edmonton, we have the orchid facility, and in Medicine Hat, we have the veggie facility at the moment. Side, you can see that is approximately 3 billion years ago, it split to produce that bacteria and archaea category. And then later on, 1 billion years ago, it split again and it separates us the animal, fungi, and plant. And one fun fact about this is fungi is actually quite close to animal. Most of the time, like people, you really like, oh, those are fungus, but they are very close to animal based on the phylogenetic trees in here. So the next slide is the disease triangle. When you talk to any plant pathology, the first thing they will tell you, the disease triangle. And the classic disease triangle by Scott Torf, 2017, has proposed that in order for a disease to occur, we need to have a susceptible host, the environment factors, and the plant pathogen itself has to present in these triangles. Therefore, disease will occur if all factors are there. So I created another illustration called the life form triangle. It's essentially the opposite of what disease occur. Now it's called the symbiotic relationship. In this triangle, we see that very, very similar factors as instead of susceptible host, now we have the compatible host. We have the environmental factor and the beneficial microbes. With all these factors together, we will produce a symbiotic relationship to the plant, and the plant will be accepting the communication with the microbes, and then symbiotic relationship will be occurring. Now you ask me, when you combine these two together, what you become? It becomes very complex. It is super complex when you combine these 2 triangles together, a disease and symbiotic. In this pentagon, we have the host. We have the environment factor is divided into 2 parts, which is the abiotic factor and the biotic factor, and the beneficial microbes and plant pathogens. So the dotted line is vice versa, means that the abiotic factor has either contributed to promoting the beneficial microbes to establish or killing them. So vice versa, it has that relationship. Similarly to biotic factor as well, it has factors that can contribute to the host beneficial microbes and either promoting plant pathogen to be much stronger to kill your beneficial microbes. So the relationship with host and beneficial microbes are usually through signaling molecule induced by, to induce the plant defense. One example is like the PGR reaction, oxygen species, synthespore, terpenoid, etc. All these chemical are like the secondary metabolite produced by the beneficial microbes that to help the host to gain that specific resistance. So when it comes to beneficial microbes and plant pathogen, in this case, what do? They recognize each other, they start killing each other. And it's a big chemical warfare. It's also secondary metabolite, penicillin, streptomycin, bacteriocin, killer toxin, and alkaloid, etc. Penicillin, where do we heard this name before?
Antibiotics.
Yes, antibiotics. Commonly used. It is produced by penicillium, one of the fungus that produce that, and streptomycin, I believe, is the streptomycin who produce an antibiotic that can kill the antimicrobial. So the next one is, let's say that I have a favorable environment that's supporting beneficial microbes. So in this case, my beneficial microbes is stronger in this case. If the beneficial microbes win, we have the disease is suppressed. And let's say pathogen win in this case, we are going back to the disease triangle. Disease will start occurring, you will start seeing that all the plant will be suffering. So why is pathogen attack susceptible host? It's not like I want to just attack anyone. The main objective is to complete the disease cycle. Like as all living things need, we have to go through the cycle. We have to promote ourselves to finish the cycle to continue the next generation again and again. So there are 4 different pathogenic lifestyle. The biotrophic, which can grow on living plant cell and tissue. The necrotrophic, which is it kill the host and then it start to degrade them. Example is like the Botrytis, Fusarium, Rhizoctonia, and stuff. Hemibiotrope, this guy can live at 2 different lifestyle. They can become first as a biotrophic and then once it established, and then it try to kill the host. Example like Phytophthora, Anthraquinose, and Fusarium in this case. And then we have another category, which is not the pathogenic version, which is the symbionts, namely the endophyte and mycorrhiza. Example in this case, Subtilis or Trichodema. So this is just an example of pathogenic fungal. How do they attack the plant? By nature opening. Stomata. Let's say that I walk through my bay, I accidentally step on one plant, that plant is injured. That is naturally opening in this case. If I have no natural opening, what can I do? I can start producing cell wall degrading enzyme, start making chemical that can start making an opening for myself. Let's say I have no natural opening and I have no enzyme. What can I do? I can use force now. I can start punching the cell wall to produce up to 8 megapascal force called episorium, which means that it punches a big hole onto the plant cell and then it opens that hole for the infection to start occurring. Compared to a human bite force, it's only 1.12 megapascal. And fungal can win us in this case, basically. The special structure that they use in here is called hostorium, and one example you can see on the left side is the picture of the hostorium, and they are growing in between the cell, and that thing can use it as a suction cup, kind of like a suction cup that suck up all the nutrients from the plant. That's how they manage to kill the plant itself. So the mode of action is they penetrate, they establish, they feed, and expand to complete their disease cycle. So for the pathogenic bacteria, bacteria on its own does not have much chance to win against the plant cell. The plant is so big, the bacteria is just tiny little guy, so the plant cell can just overcome that. So in order to win that war, they produce an army. And so you need to build an army quietly without the detection of the plant defense and recognition. That picture is from Lord of the Ring, if anyone like that. So one theory is called the quorum sensing. Quorum sensing literally means that, like say, bacteria alone, it doesn't really attack the cell. Like alone, it doesn't have any issue, but it is a big issue when it comes to a big army now. So let's say that a compound called AHL molecule, one bacteria, let's say that we have 3 AHL molecules. 10 bacteria, we will have 30 compound of that. So in order for the bacteria to know how to attack the plant, the AHL molecule has to reach certain threshold, and once they reach that threshold, then it will say, okay, time to attack. And that's how they attack the plant. And usually that bacterial symptoms are like the chlorosis, necrosis, and then they start producing antibiotics to protect themselves or kill the plant. And then they also, formation of biofilm is also a key component when it comes to bacteria, because once they form that biofilm, it's really, really hard to kill. They are basically inactive, and bactericide is basically almost impossible to kill them from that on. So the third one is the symbionts. So on the left side, we have the bacterial symbionts, and the right side is the fungal symbiont. Their mode of action are very similar. They communicate with the plant. And one big example on the second slide on the fungal, you can see the comparison between the, let's say, in a soil without fungal symbionts. You will see that the nutrients uptake is much lesser, and the communication is not that the plant will have a hard time to able to have nutrients uptake. One big thing that what beneficial fungus do is a mycorrhizal relationship with the roots. They try to communicate, they try not to kill your roots, and then they go in there and they help the plant to gain more nutrients absorption in this case. Not only just nutrients absorption, plant growth, crop productivity, soil quality and fertility, abiotic stresses tolerant, and also resistant to pathogens itself. So one good example, as mentioned before, the bacterial symbiont in this case is Bacillus subtilis, and on the right side is like the Trichoderma species can help the roots growth and stuff. So once you see the sign and symptoms of the disease, it is almost, the disease cycle is almost complete. It may be already too late to prevent and destroy the disease with microbes. So a curative spray in this case is needed to knock down the population, the pest itself. Sending sample to a lab for testing and identification of plant pathogen early is recommended throughout the growing season, or observe plant pathogen phenotypically or physically under the microscope is also good as well. So the main objective of utilization of microbes, we have a main objective in here is by creating a microbial releasing system that is compatible with crops at BEVO Farm. We also want to see that after the release, we want to see whether the establishment of the beneficial microbes in the crops and the effect of on pests and bioprogram. There are certain products that can cause issue, like Aureus mycota has some issue with that, but it's only knocked down a little bit on the insignificant amount. So secondary objective is I just gonna put in there sikit-sikit lama-lama jadi bukit. It's the proverbs from Malaysia that with time it becomes hills. So you're introducing slow and steady with a high frequency of introducing. With times, you eventually promote that beneficial microbes to grow in the rock wool or any organic matter slabs. And we are utilizing the environment and growing culture of the greenhouse by releasing insignificant amount of beneficial microbes into the rock wool. So why is BEVO doing this? I can sit down and not do anything. Why we are doing this? Why we spend so much time to do this work? If a fungal and bacterial pathogen went through a sexual cycle, or the scientific term for that is called chirogamy, recombination DNA for the bacteria, chances of the secondary pathogen to inherit a gene able to synthesize an enzyme to break down certain chemical is increased. So by using beneficial microbes, we are removing the main key factors to let the plant pathogen to mutate. One good example that I took from Nimala Devi et al. 2016 study to focus on just one particular area in India, Karnataka, and then the pathology, which is the high and to the non-pathogenic. So the red color means that it is virulent, means that the thing can attack. So this study is Fusarium and Oxysporum, to be specific. And if you look at the purple color and then counted how many of those red color is there are a lot, just at a single location. And when we look at the gel electrophoresis by the gene itself, they are all different. Most of them are different. So why is BEVO doing it is to prevent this happening. This will cost a lot more money down the line because we have to do more spray almost every week just to kill that. But that have issue. That becoming another big issue. And the more spray we do, we're introducing a pressure to the system. Now, this guy will keep mutating faster and faster. So right now, that's why we are using the microbial to try to control the population of the pests from things that we cannot see. So what is dry spray? Open to the crowd. What is dry spray?
It's not a wet spray.
Correct. It is a dry spray. There's no water in there. So that is a dry spray. So the concept the original concept is come from a question in a tissue culture. Even in a sterile environment, why are we still getting those pests or contamination? In this case, on the left side is the bacteria, on the right side is the fungal contamination. So the first question I asked myself, how does the contamination get into the plant where even the environment is so sterile? Question 2. Now, I asked back myself, could we use the life form triangle to grow bacteria, beneficial bacteria and fungus? In theory, yes. If a pathogen can get in there, why not beneficial in this case? So that was the question I asked myself. So this is the idea. This is a typical looking cucumber plant and a tissue culture. Can anyone tell me what's the similarity between these two?
They're green.
Yes, they are green.
Hydroponic.
Not quite.
Substrate and roots.
Substrate and roots.
The roots.
All right. So, greenhouse environment, question mark. Microclimates, question mark. The host is there, the plant host is there. Sterile media, rock wool, plant nutrients. The environment can be supporting the microbes. So back to the life form triangle. We have all these factors. We have the environment, we have the plant, we have the microbes. Can we start growing a beneficial microbes in this case? Short answer, yes. A microbe inoculation technique is a dry spraying method that are created by me to inoculate dry fungus spore and bacteria dust into the plant. Originally, we were using a Nature Tech mini air bug, which is a very cute looking fan thing, but because we are at the propagating stage, there are so many plants, it's actually quite slow in our opinion. But it was originally from Nature Tech mini air bug, but later then we switched it to BioBee bug flow and corporate aero breeze for releasing this method. So it is typically done on a cloudy day where UV light is at minimum, high humidity, especially after watering. When curtain and vent is fully closed, you don't want to release something that just escaped. And then it fit the concept of the life form triangle. It is a presence of a suitable environment, plant host, and beneficial microbes. So it complete the triangle itself. So in this case, we have that theory now. The theory is complete. We're able to grow our microbes. So we are basically just utilizing the abiotic environment that are suitable to culture the fungi and bacteria. So that is just a simple illustration on we have the fan. It's blowing the dust. So it's a dust blowing method. In this case, this is an example that I took. It's around 4:30 p.m. at the pepper crops. We see that the carrier that I'm using in this case is the vermiculite inoculated with fungal and bacterial dust. If you look under the microscope, you can see that a vermiculite, the surface of a vermiculite has a lot of like those indentation groove. That is actually the perfect stage for the spore, because spores are so small. Those are the perfect area that the spore can go in there and stay in there. And then we release that. And before application and after application, we can see that the huge difference that the amount of dust, and then before application, there's not much. So touch base on health and safety. Like it's not, because this is the stuff that we've never done it before, and it is dangerous by exposing to so high amount of bacteria and fungal in this case. So in order to protect yourself, it's not on label too. We release it without looking at the label, like, oh, maybe we can grow fungal this way. So from my perspective, I did try myself without mask one time. Don't do that, please. It is dangerous. You get sore throat and headache later, but please don't do that. Normal mask is good, like those spraying masks. A dust mask is good enough, and then a goggle would be recommended for doing this kind of work because it's so dusty, and by exposing to such high concentration of bacteria and fungal, it will make you sick. So please be careful if you really use this method. So this is just an example of the before application and after application of the cucumber rock wool. You can see that on the left side is without application, and the right side is the application with the vermiculite. You can see that how evenly spread the spore or the vermiculite on top of the surface of the cucumbers. When we apply all this bio, how do we find them? Can we just see them? Short answer is no, I can't see them. Through the eye, I can't see anything on the water. They are transparent. So in order to find them, we need tools. The tools that we use in here is a microscope, a standard microscope. So in BEVO, we do water sampling. And in order to check whether we have pests or bios in our water, we have to use a method called reverse dilution method, which means that you are trying to get stuff. It's easy to dilute stuff, but it's really, really hard to find those things in like extremely dilution condition. So I took a method from a CTAP DNA extraction method to use on this water sampling technique. So it's a method that created by me to check water quality using a centrifuge force. Remember at one of the slides I mentioned that the bacterial cell has density and mass? This is why we need this centrifuge force to collect them. So we spin them at 5,400 RPM and 7 to 10 seconds to collect all the microbes from the water. And then we discard 90% of the top surface of the water. So we collect just that 10%, use that for our sampling. And then, so this is the example of a hemocytometer. A hemocytometer is used in the health industry to count your blood cell. We pathologists use it to count spores in this case. You can convert to different units based on the population on the scale itself. And the smaller scales is the 50 micrometer. It is a very useful scale that you can use it for disease and pest identification, because certain pests and disease, they do have a unique shape and sizes. So once it falls in that, you can like almost 50% sure that it is that category of the genus. Just comparison with human cell like RBC, red blood cell, yeast, sperm, it fall in that category, that the size comparisons. So in pepper, the fun part, the pepper. I see quite a lot of pepper growing in here. I see. The objective is to establish our beneficial microbes specifically on the roots of the pepper. With healthy roots, your plant will be already fighting 50% of the war. We want to control the disease caused by Fusarium, Pythium, Phytophthora, and etc. We want to reduce the chemical spray, preventatively utilize. Also, at the same time, we do utilizing beneficial mites, parasitoid, predatory insects, to control our thrip and aphids populations. So the microbes used in the pepper crop since last October, I believe, October, to 2025 January is Rhapsody and Serenade. Those are the Bacillus subtilis products. One keynote in here is they need oxygen. If the water has no oxygen, they won't able to survive. It has antimicrobial property against pathogenic microbes. The range is pretty high, 15 to 37 degree. They can last pretty high. It's the optimum. They can actually go down to much lower, but this is like the optimum temperature for them to doubling that rate. And then the doubling rate in this case is every 20 minutes. So from 1 to 2 in 20 minutes, 2 to 4 in another 20 minutes. So it, with the best condition, the optimum, full of foods, full of support, they can doubling their rate every 20 minutes in this case. In lab is about 20 minutes. The other product is like the double nickel biotuck, the Bacillus amyloliquefacin, also need oxygen. It also has the antimicrobial against plant pathogens. This range are big, 5.2 to 57.7 degree, huge range. And the doubling rate is 3.5 hour. It's much longer, but also at the same time, it has huge range of temperature that can stand in the water. The third one that we use is lifeguard mycoides, Bacillus mycoides. This guy can survive without oxygen. So we do have all these products that go into the pepper this year that have support of microbes that need oxygen and also without oxygen at the same time. And it can survive with or like I said, with or without oxygen. Ranging temperature is 10 to 40 degree and the doubling rate is 28.7 minutes. And other product is commonly known in the industry, Botanicard, Bioceres, Mycotal, Richie Plus, Aspergillo in this case is all the stuff that went into the pepper this year. So now you ask me, Daniel, how do I introduce oxygen into the, like my water has no oxygen. Well, first measure, we have to measure that oxygen level first. Second is through hydrogen peroxide. So one of the benefit that using hydrogen peroxide in this case is once it oxidizes, it actually break into two molecules of water and oxygen. So you can actually implement to your water sanitation protocol that if you are using products like Rhapsody, Serenade, or Double Nickel, oxygen in this case could be potentially good enough. But at the same time, because hydrogen peroxide can kill them, so this is the big issue, whether you want to increase your hydrogen peroxide level to a level that can kill everything or just sufficient to defend the plant. So it's that sweet spot is needed to do a lot of trial, need to test it out before we can like just introducing hydrogen peroxide directly into the crops. So this is our pepper this year. It's looking amazing and it looks shiny and nice. This is a picture I took, and we do have one photo that I took one time, and I was hiding inside the pepper crop. It's so nice that I can actually hide inside there. So no one would know where am I. So on the right side is the green stuff. Guess what is this guy? Trichoderma. It is trichoderma directly growing on the, it can grow saprophytically. We can directly see that microbes actually growing saprophytically on the pepper sticks alone this year. It is kind of amazing to see that the effects of the dry spray that can lead down the pathway that able to support the beneficial microbes growing directly on the crops. So BEVO pepper IPM. This is just 2-year data. I started 2 years ago, so this is the data that I collected. We used Pervicure and Belief 2 years ago, and this is the new one that I applied this year. And the orange line is the timeline of the growing timeline at BEVO from sowing up to shipping. And in between that, we have the transplanting and spacing technique did by the grower at specific time. And blue box in this case is dry spray inoculation. The dark blue box is the original plan for the drenching for Pervicure and spray with Belief success at the end of the shipping, before the spacing shipping. And then the brown box in this case is drenching. At BEVO, we drench through our flood floor. So we inoculate our mixing tank with bio. We flood the floor with those products. Green box in this case is macroorganism, which means that the all this IPM group, they supply all these beneficial mites and or any other aphids or thrips control is in this case. So I'm not gonna just go through every single thing. Feel free to take photo of all the products. I believe TerraLink has most of the product in here. And those are the products that we implement in this case this year for our pepper growing season. So the fun part, we apply all these things. How do we see them? Now this is our microscope work come in handy. This is the example of trichodema pepper in 2024, 2025. We see that in our water sample, we collected a lot of stuff. Things that never seen before. We saw that after zooming in, we can actually see that those little tiny dots are actually trichodema. This is amazing. This say good news to us. We're able to grow trichodema directly into the rock wool. And you can see that the algae is there, protozoa is there, rock wool fiber is there, all the component from the microscope is there. Things that I haven't seen yet, fusarium. Good news for us. Good news for us. In this picture, we actually haven't seen a fusarium in here at all, which is good news. But the amount of concentration of spore in this case is high, extremely high. And the water sample that came back this year is just amazing. It's the picture that I took it from before. You can see that huge water sample difference. Like, it looks empty on the left side. And on the right side is full of life. The water is not, in this case, I wouldn't say this water is clean. It's full of microbes. It's dirty. It's full of microbes. I don't want to drink this water. Maybe I would drink that water, not this water. But yeah, it's considered, I would say, considered dirty, and it's full of life, of microbes that is growing in there. Not only trichodema that we observed this year, we also see that pepper, we also see it is actually quite hard to see this product directly working on the insects. Under certain conditions, most of the time you don't see this puffy looking shape sporulation. So we saw that we actually had shawfly infested with the entomopathogenic fungus by Burera bassiana. And then I said, yeah, what is this puffy stuff? So anything that looks odd, I will start taking that into the lab and observe them under a microscope. So I cut it open, and I see that, oh, some weird thing going on the surface of the shawfly. And then I found that it's Burera bassiana. So I compare it with Sarah Elizabeth at 2014. Is there a huge similarity in this case? Huge. Almost the same thing that it appeared. And we know that exactly that by the time it's the Burera bassiana is doing its work in the system. Note that pepper we only apply twice. Throughout the 40 to 50 days period, we only applied twice dry spray on this product, Burera bassiana. Not only Burera bassiana we also found another product, Langley muscarium, which is mycotal products on shawfly and fungus net on the pepper leaf surface. So another thing is growing. Now I need to look under the microscope. And under the microscope, we found that our Langley is produced in the system itself, without even applying extra stuff. Is already in the system that killing all extra stuff that is probably dead. And and then I compared it to a 2000 study, Langley muscarium, and we found that the structure are almost alike. So in this case, I have confident to say that we have the establishment of the entomopathogenic fungal directly on the pest, but in this case is like those nuisance shawfly, which is good for us. So observation on pepper 2023 to 2025. From a pest perspective, we see that significantly reduced in pest pressure overall in 2024 and 2005 winter season at BEVO, especially thrip and aphids during the pepper season crops. On October, February, we compare to that before. We only sprayed twice this year with belief and success because of aphids and high pressure of aphids and on a particular variety called Alzamora. And so we sprayed that particular one. There's one customer received a spray for that, but most of the Alzheimer's from other customer did not get the spray. Then success on the Alberta customer do get a success spray one time as well. And then we do the dry spray method is extremely compatible with the beneficial insects and mites. Although one study did mention that showed that Bevera bassiana does have small impact on Persimilus in this case. But in this case, we are not using Persimilus, so I'm okay. It's just a small, tiny effect on them. Like it's not like wipe out the whole population kind of level, but it has impact. But the study is kind of interesting. So they did the Bevera bassiana on the spider mite. They spray for spider mite, and Persimilus has to eat that spider mite that infected with Bevera, and then it caused the casualty from there. So it's like quite a long cycle before they reach them. So indirectly, yes, a little bit. And on the right side, you can see that this is a picture that I took example for a healthy Fusarium spore species versus the one on the top. Do you see that it doesn't look good? It looks like the surface is full of holes, it is leaky, it is not healthy. What contributed that? I don't know. I do not have the answer for that, what contributed, but I for sure know that we introduced something different this year, that it causes this observation. Not only found a few, but most recent Fusarium in our water sample, it's all having that kind of observation. It is like transparently looking, it is not healthy, and it looks good. From my perspective as a plant pathologist, I think that something is killing them. So it's good. Without spraying and stuff, that's very, very good. So let's jump to cucumber. Objective is to control disease caused by Fusarium, PTM, and Phytophthora again. We want to promote overall growth with using Bacillus species this time. Microbe used in this cucumber crop this year is we changed it to a product called Biomicrobes number 8. It has 8 different species of Bacillus product inside the mixture, and we apply it through drenching in our system to promote the overall growth of the cucumber. And other one is just the same, similarly to what we see before the Botanica, Bioceres, Mycota, and etc. So BEVO cucumber IPM, similarly timeline, we see that from sowing to shipping, and this time we include a spray. We have M52 spray in between that during spacing and shipping as a preventative to knock down any pest that if we observe any. But it's a good because it's also a microbes in this case. And you see that similarly to what pepper is, except the red box is the wet spray that we done for the crop itself. So observation on cucumber. Remember one of the slide that I applied the dry spray directly on top of the pepper blocks? This is what happened. Something is growing. Something unnaturally thinks it's growing. A lot of fungal. We thought that it was a penicillium, but no, in this case it's actually a product that we apply. A product called Pre-Stop. Glycadium, formerly known as Glycadium. The new name is called Colonel Stacy Rose now. So I said that, oh shit, we have an infestation of fungal now. Oh no, what happened? So Heidi was like, it's full of microbes in here. I said, is it good or bad? I said, I don't know. so I have to look under the microscope. And we found that on top of the surface, we found that it has pre-stop products directly growing on the cucumber blocks itself. So I took it and then I compared it with the reference. One reference is the mycelia sporulation stage. You can see that in 2016 compared to one of the things that I took and found similarity. And the spore in the water is in the water. And this is another example that we see that, not we, but one of the study in 2000, quite recent study that we see that the microparasitism on pre-stop on Rhizoctonia. It's another one common disease in cucumber, Rhizoctonia wilt. We see that this product does work pretty well. So this is just a video of how things are moving. So things are moving in there. So it's looking healthy. My water sample is looking really nice in this case, like full of life, full of bacteria, full of pre-stop, full of, in this case I don't see a trichoderma unfortunately, but yeah. So this is another comparison between, this is directly from, so I said that, is it really pre-stop? I need to confirm again. So I took a little bit of a spore directly from the package itself to compare side by side. And found out that, yeah, the shape and sizes fit exactly the same. So pre-stop is growing in our rock wool right now. So I'm not convinced. So I took another picture. I'm not convinced that whether it's a pre-stop in there. So I took another picture, whether it's a pre-stop or not. So in this case, it's a fresh sample of the root cell directly from the rock wool itself. So we can see that in this picture, we see that cucumber root cell, and then we have the root hair cell directly outside from the roots. We see that we have fusarium in here. We also see that we have colonostasis, the pre-stop product is also in there. We also see that we have trichoderma spore around. This is fresh sample directly from the roots. So let's say I play the video. You see those tiny dots that is moving? It's kind of hard to see, but it is moving. Without dilution, this is the amount of bacteria growing on top of the surface of a root cell. The amount of bacteria. Imagine we have like 20, 30, 50 million of bacteria in this system. Try to fight just one little guy. Sad for fusarium. We have like 3 different bio that try to control one disease at the moment. So in this case, hey Daniel, can I apply all this bio together? Yes and no. Yes and no. To be specific, a short answer I would say yes. A long answer I would say no, because sometimes they say that all these products, they try to kill each other. All these fungal and bacteria, they have antimicrobial property. They can start recognizing each other and then they start attacking. But it's fun. There's one study that I read up. It's kind of funny. When they have a pathogen in the water, they start working together now. So it's like, let's stop this meaningless fight. Let's focus on the other guy first. Let's bully that guy. So it's that, in this case, we can see that it represents the whole ecosystem in the root cell that try to target one bad guy in this case. And amazing. We see that most of the stuff that we apply using pre-stop, we're using the bacillus product, we used all these trichoderma. Using dry spray is in there, magically. They just grow with the plant. Now we jump to the different topic, which is the strawberry, which is a very interesting topic. I only have 2 cycles with growing with strawberry. So one thing that when it comes to strawberry is, the first thing coming to mind as a pathologist is powdery mildew. Terrible powdery mildew. I hate powdery mildew. Once it's established, never get rid of it. I have to spray almost every single week, and UV. So the objective is we try to use microbes to control disease caused by powdery mildew. This Podosphaera anphanis, Botrytis, Verticillium, Fusarium, Mycophorella ferragaria. Very hard to pronounce. And nail pest. I'm not going to pronounce that word. That's hard. Nail pest is a new disease in BC, I believe, and it's very hard to control. At the moment, I believe there are few fungicide can use to control it, but why not start with bios? Bios has all this antimicrobial property, maybe works. So we try to utilize microbes for the replacement of UVC. UVC is usually the most common practice that people try to get rid of powdery mildew. It is a good method as well. So we try to prevent as much of the wet spray at all costs, because it promotes the microclimates for the powdery mildew spore to start germinate. So this is our BEVO strawberry IPM. We used to do a lot of UVC, like I mentioned before, but very similar technique that you can see in here, like a lot of dry spray. And the key things that I am using in here is actinovate and microstop. Actinovate and microstop has streptomycin in them. And streptomycin, as I mentioned before, they have streptomycin. And streptomycin does have an effect of killing a lot of antimicrobial property and killing. One main reason that we try to put it on the crop is to stop powdery mildew from germinate onto the strawberry crops. So before shipping, they get the actinovate or microstop because those two products are on the streptomycin genus. So you can use that two products for that. So observation on strawberry 2024 to 2025, we see that Soraya cultivar is very susceptible to the disease powdery mildew. Greenhouse is the perfect environment for cultivating powdery mildew. Ultraviolet C is usually used to control the pests. So last year we see that, this year, we see that we have 0 incident of trip and aphids, whitefly and 2 spotted spider mite on the strawberry. And UVC application was not established because of, like, imagine I am dry spraying all this product into the crop. Should I use UVC? Rationally speaking, no. That's the correct answer. That we try to, yes.
Congrats.
Yes. So we try not to kill all this bio. It's like, imagine I'm trying to apply all this bio into the crop and I UVC it will kill everything. So it's not preferable method. So it kind of fight against my method. So we kind of get rid of, we're trying to get rid of that UVC totally this year. And we have 0 DSI, which is a disease severity index I use to calculate the amount of fungal pests in the system. We have 0 incident from December. We started December up to April, I believe. 4 months, no powdery mildew. And then during shipping, I took a photo. I told Heidi, I think I found powdery mildew. No. I was like, I need to see this. What happened? Like, things is getting sideways. I'm scared now. We ship powdery mildew stuff. And then I start taking one leaf, and then I try to isolate things that under the microscope and see, from my observation, that doesn't look like powdery mildew. I'm safe, like 20%, I'm like, calm. And then I try to isolate it and found that, you see those little guy, rod-shaped guy? Those are streptomycin. We found that streptomycin growing directly on top of our strawberry crops. And it is amazing. It is still alive. This product is applied almost like 5 days before shipping. And then it was just putting on my desk for approximate 1 week, dry condition without touching it, and it's still alive. Things are, so in my theory that the strawberry itself, the microclimate of the strawberry itself kind of try to feed the streptomycin in this case. It's a slow release method kind of thing. So back to the disease triangle. What do we, as a grower or as a production grower, what do we need to do? We need to break the disease triangle. We want to stop the disease. What we can do in this case is through these 3 different factors from the disease triangle that we are, from a plant perspective, we can introduce the resistant plant variety or cultivar or strong origin against pathogen or gene that can tolerate abiotic stresses in this case. Or you can do crop or cultivar rotation, which is also a good method as well. From a pathogen perspective, a water sanitation with verification, not just water sanitation. Why we say verification is we need to, it's not like we are applying all this chemical to kill everything. Is it killing everything? We don't know. We have to check under the microscope whether the pest is still there or not. And then we can introduce beneficial microbes. And then I would also do preventative chemical spray as well, but also not recommended because you are probably killing more good guy than the bad guy at this moment. We're also introducing non-virulent pathogen strain, like viruses that is not enough to cause issue, but is sufficient for the immune system to pick up, to recognize the pest. It's also a good to induce the systemic resistance. But the cheapest way to do this is actually environment. The environment itself is the easiest one to control. As a grower, you can control the humidity control, watering strategy. You don't keep watering your plant. The more frequency you water the plant means that your root has less oxygen. And when the root has less oxygen, your disease is gonna keep outbreak because of like those pests are usually like those oxygen deplete system. In this case, maybe microbe bubble, you can add some oxygen into the system. Supplying oxygen into the root zone, that's the recommendation. And altering NPK concentration. This is quite unique because you can actually play with your concentration of your EC, and that sometimes nitrogen is the key factor that sometimes pathogen use that a lot. So NPK altering is actually quite fun to play with if you have a trial at your greenhouse. The main aim is to reduce abiotic stresses as much as possible because the more stress the plant get means that the pathogen is going to attack the plant easily. Also, biochemistry to assist plant. Two products in my mind at the moment is EcoShield and Silamo. Both of them has the effect to strengthen the cell wall. Bigger cell wall, thicker cell wall, not thicker, but if the cell wall is thicker, it's harder for the pest to penetrate. Maybe they need like 9 kilopascal. Maybe next year we will see that the fungus will punch so hard that it become a 9 kilopascal record. Who knows? But yes, Silamo and EcoShield is one of the one that I would suggest as well in this system because they strengthen the cell wall, and which means is in comparison to a person trying to punch through a paper wall versus a concrete wall. Which one hurt you more? Concrete wall in this case. So the future is bright. There are many beneficial microbes in the rock wool from BEVO. We are trying to create, in future, we are also planning to create a genetic and plant pathology lab to improve our quality of pest and disease identification at BEVO. We also possible that we also have a path that develop molecular marker to identify pests genetically. We can also implement biological program into tomato. From all this crop, you probably see that I haven't introduced tomato yet because we have no plan at the moment. But yeah, we have plan. It's just that we are still deciding what to do with the tomato crop itself because it's on the hairy side. So is different, basically a different crop. And working on a beneficial microbe compatibility chart, and that will be April at the back over there. She is our new IPM tech, and then that's one of the projects that I'm gonna give her to do. Looking into different type of beneficial microbe, genetic modified virus, biochemistry, upregulating the jasmonic and salicylic acid metabolism. These two pathways has been proven to be one of the crucial pathway for the disease pathogens. So plant can utilize these two PGR to fight against disease. So who knows that in future we have some product that can utilize these two chemical that can help the plant defense. And using dry spray on bedding crop in future and to control pests, especially toward poinsettias. Poinsettia is a dirty crop, so we want to test using dry spray whether it work on these crops or not, and hydrangea. Maybe next year we will have another meeting about hydrangea and poinsettias. So conclusion, a blank canvas is a bad ecosystem. Essentially, you are welcoming the pest and disease to attack the plant. So in order for everyone to bring home today, the notes is cleaning good? If your water sample is 0 life in there, is it good? So we have to make ourselves clear that maybe clean is maybe not good. Over sanitize of the water might have issue down the line. So is clean good? That is a question for yourself to bring home today. And it is not just the beneficial microbes that we apply into the crops, but the accumulation of effort using the beneficial mites, insects, hard work from the grower, growing strategy, weather, and sanitation that contribute to the overall health of the plant. We do see that trichoderma established much better in pepper than cucumber, but that is possibly due to the number of application, slightly different application. Watering and microclimate of pepper is totally different compared to the other two crops. So we saw that trichoderma is much better in the pepper than the other two. But on the other hand, pre-stop established extremely well on cucumber blocks. So this is a very cool thing that we see that it grow directly on the crops. And streptomycet, we see that because it established pretty well. And we can only observe the beneficial microbes phenotypically at this moment. Virus disease will require a genetic test and cannot be correctly identified with the tools we have currently. The dry spray is compatible with the beneficial insects. It has zero chemical residue. However, we still separate the dry spray and insects and mites release application apart from each other, just on the safe side. So that's the reference. And then thank you once again from Corporate, Anatis, Plant Prop, BioB, Van Hood, Terraling. Thank you for the sponsorship for today's events and acknowledgement for the people around. Peter as my growing advisor, growers, Heidi and Gabriella, Jason, Nick from the ops team, Jean and the maintenance team, Shinhee from the IT support. Thank you very much. And lastly, the weather. Weather is the best thing that we have to acknowledge. Good weather, good crops. And microbes begin here at BEVO. Thank you very much.